This is a training video for Reveal Q+ D9-THC in hemp. This test monitors THC levels on site in real time during the growing and harvest seasons. This allows hemp producers to collect more data points to optimize CBD production while minimizing the risk of destroying the crop due to high levels of THC. This kit is product number 8024. The kit includes a tube with 25 test strips, 25 pre-filled sample dilution bottles, and 25 clear sample cups. Additional required materials include the Raptor Solo reader, test strip cartridge adapter, food dehydrator, decarboxylator, sample cups for extraction, 70% isopropyl alcohol, a coffee grinder, scale, 25 milliliter graduated cylinder, and 100 microliter pipette with tips or single-use polypipettes. This test is easy to use, and the results can be obtained through 4 key steps: dehydrate, decarboxylate, extract, and run. The samples collected should be from the flowering tops of the plant by cutting the top 5 to 8 inches from the main stem. Samples from one lot must be kept separate from another. After representative samples have been collected from each lot, the samples must be dehydrated. The removal of moisture from the plant is an important step to ensure reliable results. Samples from one lot must be kept separate from another. Ensuring lot samples are easily distinguished from each other, use a food dehydrator to dry down the sample material until it becomes brittle. Make sure the overall temperature does not exceed 150 degrees Fahrenheit or 66 degrees Celsius. Regardless of how many samples you are running, it's a good idea to use coffee filters and rubber bands to separate each sample in the decarboxylator. Make sure to label each sample lot to ensure proper sample identification. For this video, we'll run just one sample. Once the samples are dry and brittle, they must be put through a process called decarboxylation. This process converts all of the acid form of THC to the d9-THC form. We recommend using an Ardent FX decarboxylator for this step. To begin, remove the lid and place all the samples into the chamber. Replace the lid on the decarboxylator. Ensure that the unit is plugged in. You'll know this if a green light appears on the front panel. Press the M button until A1 mode is highlighted. Press the power button to begin the decarboxylation cycle. The light will turn red. This cycle takes approximately 1.5 to 2 hours. The light will turn green when it completes. Add each sample into the coffee grinder and grind for 10 seconds. The sample should resemble a typical coffee grind between coarse and espresso grind size. To maintain sample integrity, it's important to clean the coffee grinder between each lot sample. Using isopropyl alcohol, wipe the coffee grinder thoroughly and then dry between each lot sample. Place the sample extraction cup on the scale and tare the scale so it reads 0.0 grams. Now weigh out 1.0 gram of the ground lot sample. The d9-THC must be extracted from the lot sample using 70% isopropyl alcohol. This can be obtained from any local source, but needs to be 70% and not a higher alcohol percentage solution. Measure 25 milliliters of isopropyl alcohol into a 25 milliliter graduated cylinder. Ensure as best you can that the meniscus or bold fluid line touches the graduated cylinder's 25 milliliter mark. Pour the 25 milliliters of the isopropyl alcohol solution into the sample cup and screw the lid on. Set a timer for 1 minute and shake the sample cup back and forth during this time. Please note that if you shake the cup too vigorously, your extracted sample may leak from the sample cup. Allow the sample to settle for approximately 1 minute. pipette technique is an important factor to achieve reliable results. Some pipettes have 2 stops. The pipette featured in this video has only 1 stop. In a 2-stop pipette, the first stop is for the measurement of the sample, and the second stop is used to expel the sample when desired. When performing the pipetting step, it is important to go down only to the first stop to ensure 100 microliters of sample is conserved. Each step requires a fresh pipette tip to ensure there is no cross-contamination and no residual sample in the pipette tip influencing the dilution step. A dilution step is required to obtain a result in the range of interest. Uncap a new dilution bottle from the kit. Using a new pipette tip, pipette up 100 microliters of the extracted sample, ensuring no particulate matter is collected into the sample. Dispense the sample into the dilution bottle. If using a 2-stop pipette, ensure the sample does not go down to the second stop. Cap the dilution bottle and invert the bottle 10 times to mix the diluted sample. Retrieve a new sample cup from the kit. Using a new pipette tip, draw up 100 microliters of the diluted sample and dispense the sample into the sample cup. Remove a test strip from the test strip container and replace the cap. Add the test strip device to the sample cup and set a timer for 5 minutes. While the test is running, let's set up the Raptor Solo reader. The Raptor Solo should have the endpoint track installed to accept the reusable test strip cartridge adapter. If it's not installed, use the track removal tool and replace the track. Now power on the Raptor Solo reader by pressing the power button. On the home screen, select the category Cannabinoid Q+, and then select the test THC and Hemp. Select the kit lot being used. Please note, if the wrong test kit lot is selected, the results will not be accurate. If the kit lot you are using is not available, you can scan the QR code on the tube of test strips. Enter the sample ID and press the check mark in the lower right-hand corner. At the end of the 5-minute incubation, immediately remove the test strip and add it to the test strip cartridge. Ensure that it is fully seated within the cartridge. Insert the cartridge containing the test strip into the Raptor Solo reader. The reader automatically reads the device and reports the result in percentage THC. To test additional samples, first, remove the cartridge, then press Change at the bottom of the screen to change the sample ID. Press the check mark and insert the next test strip. If testing multiple areas and lots of hemp, you can designate these different areas by going into User Preferences. Select User Preferences and select Site ID from the menu. A location ID and site ID can be entered to provide 3 levels of hierarchy to describe the sample: Sample ID, Location ID, and Site ID. Using the Raptor Data Manager, test results can be transferred to a PC or laptop by USB or Wi-Fi connectivity. This concludes the procedure for Reveal Q+ D9-THC in hemp, product number 8024.